Hi, I'm Cameron Burke, and welcome to this Kaplan MCAT Science Review for Eukaryotic Cells. This video refreshes your knowledge of the nuts and bolts of eukaryotic cells. That includes membrane-bound organelles and what their jobs are, the cytoskeleton, and how tissues are formed. The main learning goal for this video is to get a general overview of what parts of a eukaryotic cell are and what they're used for. With that in mind, by test day, you should be able to answer questions like this one about the location of mitochondria within a cell. Of all the organelles, the nucleus is arguably the most important. If our cell was a city, the nucleus is analogous to a mayor's office, where all the rules are regulated, in that it contains all the cell's genetic information and is the source of the DNA used for gene production and cellular replication, among other cellular functions. The nucleus is where DNA is located, but you can't have DNA or other molecules involved in transcription just floating around wherever they want, so there's also a nuclear envelope that surrounds the nucleus, managing what goes in and out of it. The nuclear envelope operates for the nucleus basically the same way a cell membrane does for a cell. It keeps the nucleus and the important stuff therein separated from the cellular environment and controls flow in and out from the cytoplasm. If the nucleus is the mayor's office, the mitochondria are the power plants of a cell. Mitochondria are used for respiration, which means that this is where most of the ATP that gives you energy is produced. One mitochondria is called a mitochondrion, and each mitochondrion is located in the cytoplasm of the cell. Mitochondria are special in part because they're actually made up of two enclosed membranes. The inner membrane contains the mitochondrial matrix, which is basically the bubble of space where most important mitochondrial processes happen. The inner membrane describes the part of a mitochondrion that envelops the matrix. Mitochondria have their own DNA separate from the genomes of the cells they inhabit. This mitochondrial DNA is located in the mitochondrial matrix in the same way that cellular DNA is located in the center of a nucleus. One thing that the MCAT likes to test on is the fact that mitochondrial DNA comes only from your mother, so it can be traced matrilineally. The molecular complex is the team of molecules that do work in the mitochondria. Generally, this means producing ATP, which happens mostly in the matrix and inner membrane, before ATP is moved out of the mitochondria and into the cytoplasm, so it can be used by the cell or moved to other parts of the body and burned for energy. Mitochondria make energy, but that's not the only thing cells need. For other cellular products, you can look to the endoplasmic reticulum. As far as organelles go, what the endoplasmic reticulum is and where it's located is prime test day knowledge because those facts can seem confusing. But not to worry. For the MCAT, you can think of the endoplasmic reticulum as a very special part of the nuclear envelope that assists with making proteins and lipids. The endoplasmic reticulum is the factory in our cell city. Earlier we reviewed that the cell nucleus has a nuclear envelope to help it manage DNA products. Part of that management is accomplished by the endoplasmic reticulum, which is a membrane that's literally attached to the nuclear envelope, making it easy to always locate and label. There are two types of endoplasmic reticulum you need to care about. The first is rough endoplasmic reticulum. Rough ER is called rough because it has a lot of ribosomes stuck all over it to help with making proteins, which makes it look all bumpy. The second type is smooth endoplasmic reticulum, which makes lipids and detoxifies the cell. Detoxification aside, when you think of endoplasmic reticulum, creation of gene products like proteins and lipids is what should first come to mind. Once the cell products are made in our endoplasmic reticulum factory, they get sent to the Golgi apparatus. The Golgi apparatus is our city's post office. Once the endoplasmic reticulum is done making proteins and lipids, it sends them directly to the Golgi apparatus. When they get there, they get repackaged into a form more suitable to be moved around the cell or out of the cell, and then the little vesicles are shipped off to where they need to go. We just covered how the Golgi apparatus fits into our cellular community, but it's worth looking at it as an individual organelle in depth as well. The Golgi apparatus needs a lot of surface area for receiving molecules from the endoplasmic reticulum and then sending them back out once they've been packaged. This is created by having cisternae, which are basically just folds that create space for molecules to land on. The lumen, or the space inside the Golgi apparatus, is the business section where proteins and fats are modified to make them active and transportable. Hopefully you've noticed a theme here. The innermost space of the Golgi, like the innermost spaces of the nucleus and mitochondria, is where the punchline of its main function is. Each Golgi apparatus has two sides to manage traffic. The cis face is where vesicles from the endoplasmic reticulum need to come in, and the trans face is the side that releases vesicles once they've been modified so they can get out there and do their jobs. At this point in our review, we've gotten into most of the scenic parts of our little cell organelle community. Unfortunately for lysosomes, they actually have a pretty unglamorous job most of the time. Lysosomes can be thought of as the waste management of a cell. They're wrapped in a membrane to separate them from the rest of the cell because they're filled with acidic lysozymes. Their main job is to break down cellular trash, including worn-out proteins or dysfunctional, used-up organelles, as well as captured viruses and bacteria from the immune system, and then put that broken-down garbage outside of the cell. In addition to taking out the cell's trash by burning it up in acid, lysozymes can also be called upon to rupture when the cells need to die. When that time comes, the membrane ruptures, and the acid inside the lysosome burns up the entire cell, resulting in apoptosis. Definitely not as nice as what the other organelles do, but it is really, really important to both remove cellular trash and to have a cell die when it's supposed to, or else you have cancer. Lysosomes aren't the only organelles who can be used to prevent disaster. Another important type of organelle to care about is peroxisomes, so named because they're full of peroxide. These guys are basically the food pantries of our cell, and they stop you from starving when you get too hungry, because their main job is to break down fats. They're especially important when you've been hungry for a while and have used up all your non-fat sources of energy. Peroxisomes are found in every type of eukaryotic cell, because every cell needs a backup plan for when it runs out of food, and that's what these guys provide. Fatty acids that have been eaten recently or stored in fat cells enter the peroxisome, where hydrogen peroxide's waiting for it. The peroxide attacks the fatty acid tails to disrupt their bonds to the glycerol backbone, which creates acetyl-CoA. This can then be shipped out of the cell and used as fuel when sugar is low. In the same way that eukaryotic cells need peroxisomes to fight off hunger, each eukaryotic cell also needs a cytoskeleton to give it some sort of shape and structure. Unlike our previous organelles, the cytoskeletons on the outside of the cell rather than submerged in the cytoplasm. The cytoskeleton can actually do a lot of sweet things, but its main job is to give the cell some sturdiness and shape so it doesn't fall apart. Different types of cells use their cytoskeletons for different tasks, but they're all generally made of 3 types of materials. The first of these that you might need to know about for test day are microfilaments, which are made up of actin. Microfilaments have many jobs, but they're primarily responsible for acting in a structural capacity, so a cell can resist force, and it also provides some sort of protection to the inside of the cell. Additionally, actin microfilaments can act like roads for transport proteins to carry stuff on. Microfilaments are generally tougher than what you'd find on the inside of the cell, and it's important to have protection. But because it's so sturdy, actin doesn't really allow for movement. That's where microtubules come in. Microtubules are typically hollow, unlike microfilaments. They're usually made of tubulin proteins that are bound together to create movable cilia and flagella as opposed to providing structure. Microtubules are cool because they work rather like a treadmill. The proteins in microtubules polymerize or get assembled in the direction the cell needs to move, and depolymerize in the opposing direction to allow free tubulin to be added onto the front of the filament. Without free tubulin, movement isn't really possible. So microtubules get built up and broken down very often. This makes them the most flexible of our 3 cytoskeleton materials. Finally, intermediate filaments are really fibrous and are composed of filamentous proteins. These are heavily represented in keratinous cells, like those in your hair or fingernails, and they're super tough and usually pretty sturdy in terms of tensile strength. This is why pieces of hair can be stretched and fingernails can be bent, because they're mostly made of keratin, so they're pretty flexible, but also nice and durable. We need all these different materials in our cytoskeletons because cells have different jobs. Cells who have the same job tend to band together to form different types of tissues. Epithelial tissue, usually found in places that are exposed to the outside environment, like your skin or lungs, keep pathogens out, among other important things. Connective tissues keep all your body parts together. This is how your bones are connected to each other under your muscles, and why your organs aren't jangling around in there. Muscle tissues have a lot of microfilaments in them to allow for contracting and relaxing, which is what lets you move around when all your cells are working together. Nervous tissue is what's responsible for sending the message that you should move through your nervous system and generally tells your body and cells what to do. All the larger functions these tissues perform is super important, but they would never be able to operate without each individual eukaryotic cell joining together with its buddies to create the tissues, and then collectively using all their organelles to make their cellular products and get their jobs done. You've reached the end of the content for this video, so now it's time to practice and see how far you've come with the learning goal on some questions. The first one addresses the location of mitochondria. If you picked B, you're right on track. Mitochondria are located in the cytoplasm. For this question, it's important to recognize that one of these things is not like the other. Of the possible answer choices, cytoplasm is the only one that's not a part inside the mitochondria itself. Recognizing the odd answer out is a great general test day strategy and can also get you the right answer for this specific question. The next question deals with what you've picked up about microtubules. B is the right answer for this question. If microtubules can't depolymerize, it means they can't use free tubulin to go where they want to go. With no free tubulin, mitotic spindles can't move, and new microtubules can't be built. Roman numeral 3 is incorrect. Remember that our question asks us how the drugs would affect cancer. Cancer does not grow or spread using cilia and flagella, so a cancer cell's potential to form either of them is irrelevant. To recap, there are three main components involved in the physical jobs eukaryotic cells do. The first component is the membrane-bound organelles group, which includes all the organelles contained inside the cytoplasm within a cell that help produce cell products and remove cellular waste. The second component is the cytoskeleton, which is what gives cells shape, protection, and the ability to move cellular products around and to other parts of the body. Finally, the third component of eukaryotic cell function is tissue formation, which is what happens when similar cells team up together to get large-scale biological processes done using the molecular products created by each individual cell's organelles. Thanks so much for watching this Kaplan MCAT science review about eukaryotic cells. Hopefully it was useful to you and will be helpful if you get questions on this topic when you take the MCAT.